You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast, and I have Gail McIntyre. She's the Chief Scientific Officer at Aravive, ARAVIVE.com. So Gail, thanks for coming. I appreciate it.
Thank you.
So tell me, what's the premise of Aravive? What's the company about?
Well, I think, and hopefully you'll appreciate, we have very unique technology. It's clever technology that was licensed from Stanford University. And unlike other companies that are targeting cell surface receptors, we actually have a technology that targets a ligand, and it's the sole ligand that binds to AXL, which is the tyrosine kinase receptor found on the cell surface. So again, instead of targeting the actual cell surface receptor, we target the Gas6, which is the ligand, and we prevent that Gas6 from binding to AXL, the receptor, which then means that you don't have all the downstream activation associated with the AXL activity. So very unique, and the technology involved doing some site-directed mutagenesis to increase the affinity of our protein, which is basically an AXL decoy type protein, and that's why it mops up the ligand. And by
a couple of questions in here, it's a little technical. What is a ligand versus a receptor?
Sure. So a receptor there are many different types of receptors that are present on the cell surface, and they bind to a protein. And proteins that bind to the cell surface receptor are ligands. So in the case of this particular pathway I'm discussing, Gas6 and AXL, Gas6 is a soluble protein that binds to AXL, which is a cell surface protein. And when Gas6 binds to AXL, it basically activates AXL. And the activation in the case of AXL actually ends up causing the cell to migrate, i.e. move, to invade. The cells lose their epithelial type phenotype, and that's just, and they become more mesenchymal, and that's a bit of a fancy way of saying that the cells are very aggressive and invasive, and they actually just grow into other tissues without any, sense of contact inhibition. So definitely, basically can become a cancer type cell. So
by activating this AXL receptor, it's just one of is it the predominant way, the only way in which certain cells become cancerous, or just is it one of the ways, but an important step towards that path?
Yeah, it's probably one of the ways, but in many types of cancers, predominantly resistant type cancers, late stage cancers, metastatic cancers, it's probably a pretty important pathway given the research that we've done. So, for example, in the case of platinum-resistant ovarian cancer, which is the indication that we're studying first, probably more than 95% of these patients' tumors actually are highly AXL positive. In clear cell renal cell carcinoma, we know that's about 95% of the patient's tumors are very highly AXL positive and basically Gas6 AXL driven tumors. Some of your earlier stage tumors, your again more epithelial type tumors, ones that sort of grow in place and are not metastatic. That's the key word. Once these tumors become metastatic, they're based on our research, they're going to be very highly Gas6 AXL driven. But the ones that are not metastatic yet are probably not. AXL's probably not a large player in the process.
Where is this activation happening? Is it inside of a cell? The oncogenes are expressing and they're causing the cell to turn on this receptor or make it available to this interaction? Or where is this? How does this interaction happen when it doesn't happen normally?
Yeah, so there is some normal processes that rely on this type of pathway, and it would involve migration and movement of white cells to inflamed areas. But one of the interesting things is there's no mutation involved in either, or that we know of, in Gas6 or AXL that is affected. It's not a tumor. It's not an oncogene. It's not a tumor suppressor gene. So it is a protein that's probably present on a lot of cells. It's just not overly expressed, and the cells aren't really dependent on the process. And one thing that would be helpful, I guess, to understand is that this particular pathway and the AXL Gas6 protein targets actually came from what they call a lethal screen, and it was a VHL lethal screen in Stanford. And basically what Stanford found is under less than ideal conditions, so hypoxic conditions or low nutrients or low pH. So these are conditions that your cells they're not happy with, and so under normal conditions they won't thrive. But those less than ideal conditions will cause an upregulation in AXL, and this pathway allows cells to survive under less than ideal conditions. So does that help make some sense?
Yeah, no, that helps. So you said hypoxia would cause it, so like in a tumor microenvironment, I can see there would be hypoxia. But what you said also a lack of nutrients. What kind of nutrients? I thought that a lot of cancer cells have like damaged mitochondria, and they don't respire in the same way normal cells do.
Yeah, I'm not sure. I mean, glucose, for example. So one of the areas that we're pursuing is kidney fibrosis, and so the Gas6 actually acts as a mitogen for mesangial cells, which are cells that are in the kidney, and they're very important to the structure of the kidney. And when these cells take over and start growing, in patients who have IgA nephropathy, that the cells aren't seeing a lot of like glucose or a lot of nutrients, and so that would be one example of a nutrient deficiency.
Okay. So this protein gets expressed a lot more frequently on the surface of the cell, and then just opportunistically, that means it's going to be bound or activated a lot more often and cause the cell to grow out of control.
Yes, it can. Yeah, definitely. And one thing, you brought up a good point that I wanted to elaborate on, the tumor microenvironment. So Gas6 is actually secreted by the tumor. So it's a way the tumor has for making sure that it survives. But the tumor actually can also secrete growth factors, some of the interleukins, that then have an impact on the white cells in the tumor microenvironment, and that causes the cells in the tumor microenvironment to secrete Gas6, which then feed the cell. So it's sort of a double whammy. So it's a paracrine autocrine type of protein, and seems to be there to just make sure that that cell survives under less than ideal conditions. And in the case of a cancer cell, it lets it basically go crazy.
So can you recap what activates when this protein is expressed on the surface of the cell? Does it require it to activate it to then tell the cell, yes, okay, go ahead and start dividing more frequently than you normally were?
It doesn't have a huge effect on cell division. Again, the impact is more on invasion and migration.
Oh, you mean, okay, so if I have a liver cell and somehow it migrates near my pancreas, it normally won't sit there and start dividing. It somehow gets a signal from the ambient that says, this is not your place, move on.
Right. Or it'll have a finite
life cycle. But that's not
cycle.
happening.
Right, exactly. Yeah, no, it continues. the cells continue. They don't get those signals to stop dividing. They're not, for example, contact inhibited, and so they'll keep growing over everything.
Inhibition?
So in normal cells, if you were to take a bunch of normal cells and plate them, fibroblasts, for example, you look at the plate, you see them all, the cells sort of form what you call a monolayer, and they lay down right next to each other, and the cells will grow up towards each other, but they won't grow over each other. In a cancer cell, if you take a cell that's no longer contact inhibited, you'll look in a petri dish, and you'll see these cells growing all on top of each other. They form what's called foci. So they're not contact inhibited, and that's one of the processes the tumor has in terms of just totally migrating, invading, and destroying healthy tissue.
How about for the other tumor cells itself? Is this such a does it make the cells so competitive, so aggressive that they don't care? They'll just block out every other cell, including the tumor cells themselves? Is that why you get hypoxia, or is that I can see like with normal tissue, there's a mutualism there. The cells work together. They also compete for resources, but they work together. But with cancer cells, it sounds like they're just so aggressive that they don't care about other tissues. They invade them. But are they aggressive to the point where they'll even like hurt their own selves, or are they still loyal to, I guess for lack of a better word, other tumor cells?
Yeah, I don't actually know the answer to that. I would think it's the former, not the latter. And not to be too anecdotal here, but I don't know, in the old days, I don't know if you heard when, people would say, oh, they took out her primary tumor, that, that's it. She's going to be in trouble. Because some of the tumors do secrete growth factors that keep parts of the tumor in check. So in terms of the Mets, though, once you get to metastatic disease, the tumor cells, they just grow all over the place. I don't think there's any loyalty.
Okay, so again, to recap one more time. So this protein's being expressed a lot on the surface of these cells, and that's a signal to you, okay, if we could bind up these proteins and do not allow them to interact with the outside environment, that would keep the cell from being aggressive and going places it's not supposed to go. Is that the theory behind this?
Yes, yeah.
What specifically is being expressed? What's the name of it on the outside surface of the cell, and then what's binding to it? When that binding happens, then what happens?
So the AXL receptor, which is a tyrosine kinase receptor, is what's expressed on the surface of the cell, and it can be upregulated, and there can be a lot of it expressed on tumor cells, especially resistant tumor cells. And Gas6 is a soluble protein that binds to that AXL. Is that what you were asking?
Yep. And once that binding occurs, what happens then? Is that a permanent binding or a temporary one?
No, it's not permanent. What typically happens is the first step, for example, is phosphorylation of AXL, that receptor protein, and then a lot of activation of a lot of downstream proteins. So, for example, you'll see increases in TGF beta, PDGF beta, AKT, a lot of proteins that are very well known to be involved in cancer and fibrosis and invasion and migration specifically.
So you want to cause more binding or less binding? You want to inhibit this binding so that you don't get
I want to inhibit it, yeah.
Right.
So what our technology does is inhibit it. And, you can, just by making a protein that's similar to the AXL protein that's on the cell surface to try and compete off Gas6, the ligand, to make sure it doesn't bind, it's not enough. There's such a high attraction between the AXL cell surface protein and that Gas6 ligand that it's difficult to compete off. So you couldn't produce an antibody. A lot of people will use a monoclonal antibody to try and interfere with a protein-protein interaction. But monoclonal antibodies actually typically can't be produced at the affinity that naturally occurs between AXL and Gas6. So what Stanford did is, again, they looked at the binding between that AXL cell surface protein and its ligand Gas6. They found out where in the proteins it binds, and then they started making some mutations in the AXL protein to say, hey, can we come up with a higher affinity, so a protein that is binds to Gas6, the ligand, even more tightly than the endogenous affinity, and they were able to. So the protein that we have in the clinic binds at a 200-fold higher affinity, so much greater attraction than the wild type. So you can imagine if you put this, what we call an axial decoy protein, so it's very much like the axial cell surface receptor, only it's soluble. You flood that into the organism, whether a mouse or a human, and our protein's going to win. It's going to mop up the Gas6 in the system. And then because of the way we engineered the protein, it's going to, that combination, the Gas6 that it's mopped up, it's going to just secrete out or get rid of in the system. So the Gas6 is going to get mopped up, so it's not able to bind to axial and start all of these downstream activities, which then would lead to invasion migration. So by mopping it up, we decrease invasion and migration. And we've seen that in preclinical studies in tumor-bearing mice, where if we can get the serum Gas6 levels such they're not detectable anymore, now we see a nice anti-tumor effect.
Oh, you see, so you're trying to do two things. You're trying to bind to the axial and bind in such a way that it doesn't activate all these downstream factors and produce them, right?
No, we're binding Gas6, the ligand, so it doesn't. Yeah, so it's not. It's very unique. I mean.
So you're taking it out of circulation because you're binding with it. Okay, gotcha.
Yeah, no one else. Now we have the intellectual property, I hope, pretty much nailed down around targeting the ligand. Not a lot of other drugs have targeted the ligand. They have gone mostly for the receptor, but your point is well taken. What you said is true. If you were to target the receptor, you would have to make sure you don't activate the receptor. So, one, we had the luxury of being able to increase the affinity and actually mop up the ligand. But the other reason this works so well is Gas6 is the sole ligand to axial. So there are Gas6 can bind to some other similar proteins to axial, but these other similar proteins have redundant mechanisms. So merely taking Gas6 away from them won't stop the pathway, but taking Gas6 away will completely halt the axial signaling and all the downstream activities, which again lead to invasion, migration, and the tumor process, the metastatic process.
Is there any downside, so lowering Gas6 levels systemically? I mean, I would think that might be a downside for normal tissue growth and repair and all that. If you lower it too much,
question.
I mean, you're attempting to lower it in a local environment, but that seems to be the problem with a lot of these things. It'd be so nice if you could do it locally and not systemically. But have you observed there's any downside to doing this?
No, and there's a lot of information now we have about that, and it's public information. So, number one, there's quite a bit of knockout data out there. Are you familiar with knockout data? So you can knock out either in the mature organism or in the developing organism. You can very specifically knock out certain proteins and
right. Okay,
and then see what happens, right? What happens when you knock out axial. And what we've seen when we knock out axial, well, we haven't done it, but it's out there published, and there are a number of papers now that have done this. If you knock out Gas6 or you knock out axial, there's actually no abnormal phenotype. The animals, the embryos, they develop, and there's no lethal effect in these animals. Furthermore, there's no effect in the adult organism either. If anything, there may be a positive in that you see a decrease in thrombogenic events under certain circumstances. So there may actually be some sort of advantage. But we've also done toxicology studies. So in order to get into the clinic, you have to do GLP, good laboratory practice, tox studies to support the clinical study that you want to conduct. And we have conducted under a U.S. IND a phase 1 study in healthy volunteers. And as you might imagine, there aren't too many oncology drugs that can actually be studied in healthy volunteers. But the reason is we went in and we dosed monkeys. Their Gas6 and their axial are pretty much identical to ours. And our protein binds monkey Gas6 to the same extent that it binds human Gas6. So we did toxicology studies in monkeys, And we were able to dose monkeys at very relatively high doses. Actually, we couldn't go any higher given the concentration of our drug and how long a monkey will sit still for the infusion. And we were able to dose, for example, 150 milligrams per kg every week for 4 weeks, and we didn't see any adverse effects in our monkey studies. And in contrast, we knew that 5 milligrams per kg in a monkey suppressed GAS6 levels for that dosing interval of 1 week. So there's a 30-fold margin between what was effective in terms of suppressing the GAS6 in the monkey and the dose that we could dose in a monkey without any adverse effects. So that's unusual to have an oncology drug that has that kind of margin in the GLP tox arena. And so that allowed us to go into healthy volunteers. And because we have the serum biomarker assay, the GAS6 study, and it's a proprietary study that we have developed and validated, we were able to go into the healthy volunteer study and look at serum GAS6 levels and find out in our healthy volunteers at what dose will suppress GAS6 levels for 1 to 2 weeks. And then we took that dose. We made sure, first of all, it was safe, of course, but then that dose, which suppressed our healthy volunteer GAS6 levels over 1 to 2 weeks, is what we're now testing in our ovarian cancer study. We're testing a 10 mg per kg every other week dose. And the reason is, number one, going into healthy volunteers lets us get into, a more defined oncology study sooner. It also means we're not slowly escalating up across a lot of doses that may not be pharmacologically relevant. So we're now in the clinic in platinum-resistant ovarian cancer patients with a dose that we know suppresses GAS6 levels for the 2-week dosing interval. And given our preclinical data, as long as we see suppression of GAS6 in the little tumor-bearing mice, we have the anti-tumor effect. So we figure this is a really good way of really testing our drug.
So what happens, though, once you take GAS6 out of circulation? Do the tumors just stop metastasizing and hang out, or do they actually shrink and die because now it's another door closed to them to get nutrients and get oxygen?
Yeah, they shrink and die. So we've tested a number of preclinical models, and you use human platinum-resistant ovarian cancer cells, and they're very metastatic cells. You inject them into the peritoneum of a mouse, and they develop in the mouse much like you would the human, in that the tumors seed all throughout the peritoneum. So it's a lot like a human ovarian cancer. And when we treat these mice, what we find is that 20 to 30% of the animals actually don't have tumors anymore after 21 days. You have to obviously sacrifice the animals and open them up and look at the peritoneum and look to see if there are any tumors. The control group, i.e. the animals that didn't get our drug, have thousands of tumors. But our animals that were treated with our compound, along with, I should say, doxorubicin, actually 20-30% of the combination, actually, those animals are cured. Our drug does have single agent efficacy in that model as well, where all 10 of the animals reproducibly have minimal disease. But the combination with doxorubicin or Doxil is pretty phenomenal in that we have 30% cures.
So in the trial, does this have to be combined with chemotherapy, or is it before chemo, after chemo, or along with it?
Along with it. So we're doing it in combination. We feel like that is going to be the best benefit for our drug. Again, our drug doesn't have probably a direct anti-proliferative effect. Its effect is greatest on the metastatic processes of the tumor cells. And our preclinical data suggests that in combination with drugs that damage DNA or just generally cytotoxic, we should at least be additive to that process. And oncology drugs, or treatment is going to be combination. So we are going in combination. Our phase 2 study, so any pivotal studies, what we'll do is we'll look at the combination versus what we call a placebo arm, but it's not strictly a placebo arm. So if we want to study our drug in combination with paclitaxel, we will look at any pivotal studies. We'll look versus paclitaxel alone. Or in the case of combination with Doxil, we'll look versus Doxil alone. one really important point and something that allows us to look at the combinations is that our drug is a protein. So unlike small drugs, which are metabolized by the liver cytochrome P450 system, so enzymes in the liver, our protein's not going to use that same metabolizing system in the liver as small drugs. So we're not concerned about drug-drug interactions as you typically would think about, because our protein will just get broken down into its constituent amino acids, and it's just, we don't worry about drug-drug interactions. And because we've not seen any toxicity, at least knock on wood until this point, we're not concerned about adding on to any of the toxic effects that you see with chemo. So we just don't have the concerns about drug-drug interactions.
Oh, okay. So is this going to be used in patients that have metastatic cancer, or is it going to be used early on where there's just a primary tumor?
Really good question. So right now, given our preclinical data and where we think our drug will have the greatest benefit, we are looking at the platinum-resistant population, so the later stage population. But as you can imagine, we are talking with internally, and we're also talking with some investigators who may be interested in doing investigator-type sponsored trials, because there would be an interest in backing up and looking at earlier stage patients to prevent the metastatic process. Now, in my opinion, this is my personal opinion, I think one day, idealistically, our drug could be given in combination with, your typical chemo regimen or whatever you need to treat the primary tumor, but our drug could be given long term to see if you could prevent that metastatic process that pretty much, and it occurs in pretty much all cancers eventually, the more horrible cancers specifically. So it'd be sort of like a tamoxifen type compound where it's used more prophylactically to prevent the mets. That's not a study we're undertaking at this point, and we're a small company. That would be a very expensive study, very long-term study. I think ideally, though, that would be the study that someone would want to do with our compound.
So why focus on the later stage? Why not focus early on prevention, where it seems like, in general, a lot of things are easier? Prevention in this conversation, meaning, when there's just a primary tumor, even before chemo, maybe you wouldn't need chemo, possibly.
Well, yeah. Well, the sad thing is, again, I don't think we're going to have the dramatic effect on the primary. It is going to be on the metastatic process. But so the indication we're in now, unfortunately for the patients, the progression-free survival is 3 to 4 months. So, you don't need a large study and you don't need a long duration in order to understand if your drug works. So the first objective for us is to see if our drug's going to work in probably the most realistic setting for our drug. And because we have the preclinical data in which we tested mice, inoculated or injected with platinum-resistant human ovarian cancer cells, and we see such a dramatic effect, that's where we went in the clinic. And that's, again, unfortunately, a pretty short-term study.
What do you think is happening between the primary and the metastatic sites? Do you think that the AXL protein is being expressed more frequently, even on the primary tumor, and because of that, it's allowing those cells to divide and break off, or at least divide, and those daughter cells now migrating throughout the body and invading? Or do you think the mechanism of action is different in the primary tumor versus the metastases?
I don't know for sure, but I think you hit on an interesting point. There was at least one paper out, and I can't remember the author, a while back, that talked about exactly what you said, that the primary tumor some of the cells can increase their AXL expression. They can break off because they have AXL expressed. They're gonna migrate. They're gonna literally move, so they can break off, move, and they can set up at another site. Basically, that's your metastatic process. So that probably, that makes sense, and that probably is what's happening.
So I know these clinical trials can take a while. How long, and what stage are you at? Are you at stage 2 with the clinical trial, and when do you expect to see results, and then what's next?
So we're doing what we call our safety run-in study to the phase 2 randomized, blinded, controlled portion of the ovarian cancer study. And what we're looking at 6 patients randomized, or not randomized. It's not a randomized portion, it's a run-in. 6 patients given our drug in combination with Doxil, and 6 patients given our drug in combination with paclitaxel. We have an independent safety data committee that's meeting to review the safety PK and PD data. If they agree those data look satisfactory, then we'll open up and allow for 6 more patients in each of those cohorts. So we have told the public that by Q3 of this year, we expect to be able to announce the safety PK and PD data on the 24 patients that were involved in the phase 1B, i.e. safety run-in portion to our phase 2 study. And then we hope to initiate the phase 2 study right after that, which would be the end of this year.
Very good. How can people get more info and reach out and ask questions or maybe look at the clinical trial if they're eligible?
So our study is actually listed on clinicaltrials.gov. And if you do a search on Aravive, the company name, you'll pull up that study. And it lists the inclusion-exclusion criteria, and it lists it, at a high level, the study design.
Okay, very good. And so Aravive website and clinicaltrials.gov. So thanks for coming. I appreciate it, Gail.
Thank you.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you.